So welcome everyone. This is essentially the shorthand or the name that I will be using a lot is Data Science for the Biomedical Science Workshop. It is a workshop taught in R. If you will be so kind, there are a set of surveys that help with improving this set of materials, especially trying to figure out what you think is useful and what I can do to help provide educational materials around those things. So these surveys are around an actual IRB and is for my dissertation. But if you will be so kind, it would be great if you take the pre-workshop survey even after today, and even though it's over 2 days, and then after tomorrow, I will send out information for the video recordings and also the post-workshop survey. We have my advisor, Ann Brown, who's also on the call with Zoom and helping out with Zoom related things. She has a dual position in the biochemistry department and the university libraries. So she will be helping us with all of the other logistics and stuff behind the scenes. If you have any questions, feel free to message her, or you can also message me or direct message me. I am very used to reading chats flow through the screen as I'm teaching. So don't worry if you want to ask a question, it can go to either of us. I am the instructor. So my name is Daniel. I have a master's in public health and epidemiology. I teach data science related tools pretty regularly. And a lot of the work that I am presenting today is from my experiences from teaching, and it's all bunched up into my current dissertation project. Quick links for the lesson materials that we will be covering. All of the stuff I will be covering in today's workshop is on this website called ds4biomed.tech, where you can read the prose stuff and the code if you need a reference to go back to. If you are here for the whole bunch of series of workshops, more and more stuff will get filled in, especially for the Python stuff later on this week, and then the R part 2 stuff next week. But everything that we're covering in this workshop is already up there. I will have different slides and papers that I will be referencing or referring to during the workshop. And so you can always find the stuff for that at this Google Drive, which is ds4biomed-G drive under bit.ly. And then in general, if you get stumped with terminology in the tech world, there is a like global glossary that people are working on just to help you with identifying and defining terms. And the question is, yes, the actual GitHub repo will host all of the content for all of the courses. The website itself, or that ds4biomed.tech, is actually hosted on GitHub. And as I add more materials, it's all going to be there. Before we actually start working with data, I'll also show you how to download the data sets, regardless of today's workshop or whatever other workshop. For those of you who are remote, and if you are on a remote system and you only have one computer screen, this is one suggestion on how you can lay out your screen. The more important thing is you need an RStudio window that is essentially big enough for you to work in. And if you're on a 1080 screen and even a 720 screen, try to have it on the 1/2 of your screen. You can take the zoom window and make it a little bit more than 1/4 on the other part, and then you fill in the other 2 bits just so you have the Zoom chat and whatever notes that you're taking. So this is just a suggestion, assuming that you only have a single screen to work on. If you end up going to the website within the next couple of weeks and you end up seeing this notice, it is literally because I am doing a website update and all of the content is being updated. And the way the whole system is set up is it sort of throws the website into this page when it does that, which I try to do the website updates very late at night, but if you just so happen to stumble across it, that is probably why. And usually it'll rebuild and redeploy itself in about 15 minutes. There is stuff about I mentioned that a lot of the information from today's class is around dissertation data. And so this is a small glimpse of the research that is underlying today's workshop. And essentially, I am trying to get you exposed to learning R or Python for dealing with your data. And a lot of the motivation is around you want to be able to work with your data in its rawest state, but you also want to be able to reproduce your work. And there's sort of like a mismatch if you end up working with your data workflows in Excel because you're actually then modifying your raw data set. And we try to show you ways where you can make your data set, even if it's in Excel, more useful to other people and to yourself. A part of this work is around a set of learner personas, which you can view them on this part of the website. And essentially, learner personas are fictional characters that we have created from the survey data that we collected, and it helps me target the information that essentially you will find interesting and want to learn. If you want, you can, I read through their learner personas. We're not going to go through this, but essentially this set of materials assumes that you're either an academic, a clinician, a programmer, although if you are like an expert programmer or you program already, this might be a little too easy for you, but you can always ask questions. And then, or if you are a student, and this is all in the context of working in the biomedical sciences. So for those of you who are in this workshop, you've done the first thing of learning R or Python. It is going to be a journey as you learn this set of materials. Hopefully after today, we sort of get you over the initial hump so you have some things that you can work on, but do know that the more that you program and the more you get involved, there's going to be multiple learning curves as you start using programming as a tool for your analysis. And so one of the things that happens to everybody, there are many phases when you are working with code. The computer will do exactly what you tell it to do. So most of the time and this happens to me a lot, this is the same series of emotions that I go through when something isn't working properly. It happens to everyone, not just because you're very new to all of this. So just starting that off, I hope that you don't get too demotivated as you end up implementing and using the skills that we will cover in the next 2 days. And so another thing that I'll cover at the very end of tomorrow's class is there is a lot of resources and people that can help you after today's workshop. So don't feel like you've taken a class and then you are sort of on your own. There are a lot of online resources and communities that can help you with any of the problems that you have and continue learning. So what do we expect today? There's a lot to cover for very little time, or what do I expect at the end of the 2 days? So I don't expect you to rewrite entire R analyses from scratch. But hopefully when you are given another data set, there is a plan working forward. And then also you have the terminology to describe your data set or data problems if you end up collaborating with a statistician. So at least you have some way to talk about your data set. So for today, we have 3 hours today and 3 hours tomorrow. So today we will end up talking about how do we go from spreadsheets and setting up spreadsheets in a way that is more conducive to data pipelines, and also how we can create data pipelines. And we'll end off with just a little bit of doing descriptive calculations. And then for tomorrow, we'll build on all of the stuff that we covered today, and then go through what does it mean to clean data, how do you visualize data, and then from my MPH background, a lot of the medical questions that we want are use logistic regression. So we'll sort of show you how to run logistic regression and do the analysis from there, and also interpret the results. All right, so that is the intro slide deck to get everything started. All right, so the next thing is, and if you are in, if you like sort of just came in, we're not like actually coding right now, so don't worry too much. Like there's still time to sort of catch up. The first bit that we're gonna work on is sort of a series of thought experiments. I used to just have you like do this for real, but for time, it's sort of like not really useful. So we'll talk about spreadsheets, how we can set up spreadsheets and all of that. We will, and then I'll repost links for the necessary links as we go through. But for now, everything that you will ever need to find is on this website, ds4biomed.tech. All right, so first thought experiment. Let's say you are tasked with collecting data for a pharmacokinetic study. And usually these are frequent blood draws in an essentially like a research portion of an ICU wing, or in a research, an ICU ring in a research part of the hospital. And you have 3 patients. And you have this set of data. If you want, you can open up a spreadsheet program and then enter in this set of data, or take out a piece of paper and roughly draft this out. But I want you to either physically or mentally input this set of data in a theoretical spreadsheet program. So I'll give about a couple, I'll give you a little time to sort of just think about it, knowing that there's going to be more data for each patient, because it's going to be data collected roughly like every 10 minutes to 30 minutes for 24 hours. All right, so one thing you could have done is for each row, you essentially just put in what you collected on your note paper as you're walking and doing rounds in the hospital. So we have one person represents each row, and essentially units for blood pressure and all of that, you just entered in whatever the thing you wrote down on a piece of paper. One of the problems with this is that a lot of these numbers, or your data set that you created, actually don't have units in them. More specifically, the temperature column doesn't have units in them. For those of us in the United States, it's probably going to be Fahrenheit for the most part, but a lot of multi-site collaborations, especially if you are international, will probably not use temperature in Fahrenheit. This, at least for temperature, it's a pretty easy thing to realize what the unit is. because if you end up with a body temperature of 97 degrees C, you know that's pretty much impossible. But you don't want to have to deal with someone else putting in data into your data set, using the unit of measure for whatever they have, or maybe the thermometer, someone bumped the thermometer and it just read into Celsius mode or flipped to whatever, and you're just reading off of the wrong number because that's what you're just collecting. So the data is there, it's just sort of in a mixed unit format. Other things like pulse ox, we essentially know that that will be percentage. Respiratory rates are typically rates over a minute. Same with heart rate, it's over a minute. Blood pressure, this is a little bit problematic because our systolic and diastolic numbers are also all in one column, even though we know the unit is going to be typically millimeters of mercury. And then the other thing with time, it might be clearer if we have more units of time, but right now, I don't know if that's really 8 o'clock in the morning or in the afternoon. Are we using 24-hour clocks or 12-hour clocks, et cetera, et cetera. So one solution to this is you can just put the unit straight into the data set that you want. That is super explicit and that is okay. The only problem with doing this is if I were to, even in Excel, because it's 96.8 F, Excel will read it as a character and not an actual number. And so that ends up being a problem because there's no way to now tell, even in Excel, say, take the average of this column. So that applies to any other column where we are putting in units. One of the other solutions is you can take the name column and split it into two. This is pretty common. You'll have a column for first name and last name. Sometimes that ends up being problematic because depending on your ethnicity and the way your name is set up, it doesn't always split into proper first name, last name. But that is something that is what you can do. And part of the reason for that is we typically end up searching for names based off of one or the other. And if we put the entire name together, usually the problem with that or the hurdles we have end up being that there's no way to really work with like a last name on its own or a first name on its own if that's how we're searching. And that's the same problem with the blood pressure column because there's literally 2 units of measure inside that single cell. And so it means that we either can't just say average systolic heart blood pressure because we are now looking at 2 numbers, but it's not even registering as a number because there's a slash in there. And so that ends up being a problem as well. Talking about units, one thing you can do instead of just putting in the units as the value is put the unit information as part of the column name or the header. This way, it's really clear to anyone else if they look at the first column of your data set, what the proper unit should be. And then you don't see it in this case, but you now have the added benefit of everything in that column is a proper number. So I can now say what is the average pulse ox for all of my patients, or what is the average temperature if I didn't put in the letter F in there. So another thing you can do if you know you're going to end up with mixed units or something else, or adding some form of metadata to another column, is to really just have a separate column that is the metadata information. So one thing you can do is if you're going to record temperature, and you might notice that, you know, depending on the people in the team and what units they're most comfortable with, if things are going to be flipping between Fahrenheit and Celsius, make a separate column that's just recording what the actual unit is. Don't end up doing like, oh, if it's blank, it's the one before, like actually just go and fill it in, because now you have some form of metadata where if you need to do a temperature conversion, you can, instead of just saying like, oh, if the temperature is below 80 or in the 30s, and it's probably in Celsius. So you don't have to do like those weird heuristics. And then everything is much easier to filter. You can just say like, give me all the rows where everything's in Celsius, and then I can just go and convert them or fix it that way. So the main takeaway for that really is put one thing in each column. It is really important that if you are curating your own data set, or if you are given a data set, you should be able to point to a value and then just say like, what is the thing this is measuring? And hopefully you're only able to say one thing. We'll talk more about this problem tomorrow when we're going to fix data, or how to clean and process data, how we can deal with what happens when you put multiple things in a column. But this is one thing that, especially if you're curating your own data set, like keep this in mind. So we'll take another minute or so, and then let's say I just talked to you about that first set of readings. So here's two more readings. They essentially end up being like exactly the same because I've copy pasted things wrong. But let's say that there's two other sets of readings, 12 hours apart and then 24 hours apart. This isn't really like an actual PK study because they're usually done in like 5-minute intervals in the first like 3 hours, and then it extends. But how would you end up inputting data for multiple people, knowing that there's going to be like a lot more data points for each person? So think about how would you actually go do it if you primarily work in Excel. Like how would you actually go do this in Excel? There isn't really a wrong way to really work on all of this. Most problems can be fixed. It's sort of just the how much more work you have to do later on. So okay, so one solution is you can group up all of the data by patient, right? So this can either be in separate Excel tabs. You'll have a separate Excel tab for each person that for this research study. Maybe that's okay because there's only 3 people right now in this clinical trial experiment and not like hundreds. So you don't have to worry about hundreds of Excel tabs, and you have data across 3 separate ones. Totally fine. And assuming that all of the things I mentioned before about putting a single value in a single column still apply, there's really nothing wrong with setting up your data that way. And I believe I also flipped the, there's small mistakes with the actual thing with the slides, but there's nothing wrong with having separate people in separate tabs. Knowing that, if I were to ask the question like, oh, what is the average heart rate across all of my patients for the entire PK study, or their first reading or their last reading, you're going to have to do a little bit more work because they're in separate tabs. There's no way for you to essentially filter that set first. You have to find a way to combine all of that into a single data set before you can go and answer that question. So just knowing how you set things up, as long as you have a plan for how you're going to do this analysis later, everything's okay. So one thing that you can also do, and I see this very often, is, okay, we know that separate tabs is going to be annoying because I can't just take an average of a column. So we will just put everyone into one data set, which is fine. But then we visually put in white spaces or new lines or empty rows for us to visually separate each patient from one another. This is fine, really. The only issue with this is if you end up loading this into any piece of software that's not Excel, all of those blank rows will end up being data, and that data will say like, this is an empty value. And if your question is, give me if I'm just looking at, for example, the heart rate column, and I just give you a set of values, part of that set of values are going to be a bunch of missing values. And then now you don't know if that's a missing value because you are visually putting in blank rows to separate people in your data set, or that value was actually not collected or not collected properly, or the machine broke, so it's actually missing. So there's no way to actually go about and teasing that out. And so essentially, like when I load up data and see a whole bunch of new rows that are blank, like usually if it's like the entire row, the first step that you do is like essentially drop them. So you don't have extraneous blank rows in your data set. But it is one of those things you want to be very careful of. And it's something that happens, or even when I'm curating Excel data sets or putting it together, yeah, it's really easy to sort of like, hey, I'm going to use like this area for like scratch work and then like slowly put things in as I'm curating data set. But that is something you have to think about because your denominator, if you are taking averages, is going to be different if you include those missing values as like an actual patient reading. One thing you want to be very careful of, and this is one of those things where you definitely do not want to do, is instead of stacking your patients one on top of each other, you stack your data like next to each other. This is really common if you're working with like time series data. And more recently, this happened in the UK where essentially they had COVID counts for each county. Each row was essentially their equivalent of a county. Each column was a new day of the outbreak. And then essentially they made whole data pipelines around this whole Excel file. And then they literally ran out of columns in Excel. And everything went wrong because Excel literally ran out of columns. And so you do want to be very careful if you're tacking on new data, if you're tacking things on each column, you definitely want to be mindful of that. Having said that, maybe your initial assumption was wrong and you didn't expect that many columns, or maybe you didn't even know Excel had a column limit, just so you know that that stuff is there. But the other reason for this is that type of thing where you essentially run out of columns also happens to a lot of database systems where they have like some factor of 2 limit of how many columns you can have in a data set. And that's also because your data shouldn't be formatted that way. It should be formatted where new information is put in rows. And that's just how data should be stored. Having said that, there are ways to go from row-oriented essentially to column-oriented, and we'll show you that tomorrow. But be very mindful if you just end up tacking on data in each column. And so one thing that you can do is not have your data set put into weird white spaces. You're not tacking things on column-wise, and you just put all your patients in a single table, and then each row is just the next time observation. That's useful from just the data analysis perspective, because now I can just say, give me the average heart rate, or for each time of the day, give me the average heart rate. So now I can compare across like how the average is happening for all of my patients in each timestamp. We can do those operations, but that sometimes comes at a price where if you are curating this data set yourself, it's harder to see what's missing. Usually when you're like live taking notes for data and then trying to input them, you know, you're really just walking around with or I used to just walk around with like a clipboard and a piece of paper because I'm not entering data into the machine like in real time. And so you might end up missing something. And if you end up putting in data this way, you have to do a little bit of work to make sure that everything's input properly. The good thing is, oh, give me 1 second. The good thing is, at least in Excel, you can, you know, sort by time or last name and first name, and then you can actually go and use some of that set of features to double check your work. But that is something you need to think about. So
you're talking about this just a little bit ago, but you're saying it'd be bad to have like heart rate 1, heart rate 2, heart rate 3.
Yeah. So the question is, should I have my data set up in a way where it's like heart rate 1, heart rate 2, heart rate 3 across each column? So if you know it's going to be totally fixed, and it's just like, I'm only taking 10 readings and like, that is never going to grow. Like days of the pandemic is just going to keep growing at this point. Then it's then it's totally fine. And there's always a difference between the data in the shape of the format that makes it easier for you to input data and check that that data is correct. And then there's like the format of data that the computer needs to do the analysis. And so if it's easier for you to have it column-wise for you to input data and check your work, that's totally fine. And then tomorrow, I'll show you how you can essentially just pivot between the two views. So like you have less to worry about, like as long as you are okay manually curating your data, you have a plan for how you can do the analysis later. But that is just one thing. And that just really is, if you are storing data like longer term, you need to get data into like a long format, especially if it's like some chronological data, because there was a part where like England just didn't have like their COVID system set up because they ran out of columns. So those are things that as long as you know about, you can plan for it. So maybe like not just keep one entire spreadsheet for all of your data and just do it by weeks or by months so you don't run out of columns. But then the trade-off with that is not everything's one big data set. You have to combine them before you do the analysis, but that's fine. You're not going to run out of columns. Or so there are, as long as you know about these problems, all of these things can be addressed later on. And so that is the actual point. Like, as long as you have a plan moving forward and it's not just like, I just need to collect data and just putting it into a spreadsheet, the more you can plan on how the data can be analyzed later on. Maybe that'll inform you like, hey, I'll take the little hurdle of putting like the F next to the temperature and make a column for that, because I know that this is going to go into like, there's going to be a whole bunch of people from across the world inputting data, right? So as long as you have a plan for how you might deal with something, there's always a solution. And the better you plan this out, the less messy that solution ends up being. So just to round off the example, or the next exercise is, let's say you are running this clinical trial and there are a set of like adverse event rules. How do you go about flagging which observations need to be addressed, right? Like, let's say your blood pressure is over 140 over 90, your heart rate's like over 90 for some reason, your respiratory rate's over 20, you know, oxygen's not like being absorbed by your red blood cells, and you start to have a fever. How do you go about flagging this stuff in your actual data set? So I'll sort of just let you think about that for 30 seconds. All right, so most common solution is you have the set of rules, and you, as you are inputting them, or as you're doing your analysis, or in Excel, you can literally filter the columns by some value, et cetera, et cetera. You go about and you start color coding values. That's okay. I do this. Like, I will admit that sometimes when I am given an Excel spreadsheet, I will go do this. The only problem with doing this is, remember the first rule that I mentioned, if I give you that number, that last row, that 142, or the 101 for a heart rate, what is that 101, or what is contained in that cell? It is more than one bit of information. It is 2 bits. The first bit is the actual reading for heart rate. And then now, because you input a color, it's also saying that this is an adverse event reading. And that ends up being a problem, because if you load this in any other form like spreadsheet program, they are not going to know that that cell is red. It's just going to show up as 101. So all of that time and effort that you did to color code your values essentially is just out the window. And it's more annoying if you end up doing this manually, because like all of that manual work, and you can imagine if this happens across hundreds of readings, is not really going to be useful. And so one thing you can do, and it'll make your life and other people's lives easier, is either create another column that essentially says like there's something wrong with this row. And then you can use that information even in Excel to be like, if there's something in this row, let me just color code some set of this value. And you can set up like conditional color conditions in Excel for this. But if you can either have like some column to say like, hey, there's something wrong with that value. And then now, even though that the heart rate value might be 101 with red, there's also another corresponding column that says like, hey, there's actually something wrong with that value or that row that you might need to look further into. I'll show you how we can like sort of filter values in today's class, but if for like really more complicated like recoding problems, I'll point you to how you can recode, but the class next week will talk about functions, how you can actually like write really more complex like coding rules if you need them. And so the main takeaway from here about not using colors is like actually treat your data as data and not as a data visualization, because that's when you end up saying like, oh, I'm going to open up this Excel sheet. I just need to quickly figure out like where my problems are. Now you're essentially just saying like, I just really wish this data set was a dashboard. And so like, I don't have to like do all of these other things. I just want to see like where the problem issues are, right? So that ends up being like a problem if you end up going down that route too far, because you end up putting all of this metadata information into single cells, and then you try to load it or you give it to a statistician and they're like, okay, so like what's going on? Like I can open this in Excel. There's like 5 colors, like there's a background color and a character color. But I only see like, I only get one value and what is going on, right? So you want to be very mindful that you treat data as actual raw data. And when you feel like you are running into this world where you're treating your data as a visualization, like think about it and actually like create a visualization from your data and not just start manipulating the actual underlying data set. And then the last thing, let's say, for example, the reason why your patient's heart rate was elevated was because they essentially didn't follow directions. What do you do? So I'm not going to have you like go through this thought process and just sort of just say like, yeah, just make sure you have room for, if you are manually curating data, and this isn't data from like a sensor or something that you're reading, have a column that are essentially like just notes that you can just type in, because you're essentially trying to capture qualitative data, and it might be helpful for the next person if they're trying to figure out like why their results are weird, because this is probably a result that is an outlier, but it's an outlier because there's like something fundamentally like different about that observation versus like this is an outlier because like there's something actually went wrong during the PK study. And so all of that information can be captured in another column or some other notes field. And so the main takeaway before we slowly start diving into programming is it's totally fine that you use a spreadsheet program. A lot of times, if you end up taking an introductory programming course, people will just say like spreadsheets are terrible, never use them, et cetera, et cetera. There is a time and place for them. It's totally fine, especially it's really like the only way you can manually curate data really is some sort of spreadsheet program. Trying to manually curate data and writing that into a program is pretty tedious. And it also is harder to check that your data input is correct without writing more code to check that. So spreadsheets, totally fine. Graphical user interfaces, so GUIs, they're very convenient. So like stuff like spreadsheets. But the problem with spreadsheet programs is that they are less flexible in the amount of the type of work that you will need. And you probably, if you struggle with Excel long enough, you end up learning VBA, which is the scripting and programming language for all of Microsoft's products. But then you're down the road of programming and sort of like why you can also learn like an analytics language and just do your work that way instead of programming which buttons to click in Excel. So just realizing that where might be a good place to be doing work in Excel in a spreadsheet program and where might not be a good place is one of these initial starting points as you start working with data and programming. And it's totally okay if you only learn one thing in the next 2 days and it's like, hey, that's the thing I want to do with my Excel spreadsheet. You can totally load that in, do that one thing and save it out, and then just keep going on with what you're used to in Excel. But you slowly start building a reproducible pipeline, because if that Excel spreadsheet updates or you add more data, at least you have one way to consistently do that data transformation. All right, so what we are going to do is we are going to load up and open our studio. And if you have our studio open, and let me make this bigger. The first time you open up our studio, it's going to show you essentially 3 panels, and it'll look something like this. On the left-hand side, this is where actual code gets typed, run, and then the results get fed back to you, right? So I can do something like 3 plus 3 and use R as a calculator. Great. I can put in 3 plus 3, R captures it, calculates 6, and gives you 6. The top right panel, if we click on environment, and usually this is the one that's by default selected, as we create and load data sets into R, here we'll have the running list of everything that we created, right? So one of the nice things about R, and the last time I checked, when I worked with, I think it's either SPSS or Stata, R can load up multiple data sets. Not every single piece of statistics software lets you load up multiple data sets to combine them. They essentially say you have to combine them yourself outside. And then the only thing they're concerned with is, I need to do the analysis on this one data set. So R does allow you to see the running tally of everything that you've loaded up or created, and et cetera, et cetera. On the bottom right-hand panel, you have, by default, it will open up the files panel. And essentially this is, if you looked at this, like the way you click around your computer, this is the view of the files and folders within our studio that you can move around in. If you are trying to find or open up a really specific folder, which I will show you in a little bit, you can use this file system if you want. It's a little bit weird on Windows sometimes because you can't always like find the desktop because Windows is kind of weird that way. But if you look at the far right-hand side, there is this set of 3 dots. If you click those 3 dots, it'll actually give you something like that you are probably used to seeing. And then you can go and navigate to your desktop or whatever file or folder on your computer to load that up. Before we do get started, there is a set of things within our studio that are essentially good settings to have. And if you've never loaded up our studio, I might as well just set you off like in a better setup. So if you click on Tools and then you go to Global Options, you will be presented the options screen. By default, it'll open up to the first page, which is General and Basic. And if you scroll down, or if you look down, I have to scroll down because it's zoomed in, you're going to end up seeing this set of checkboxes. What I personally like to do is making sure that these 2 checkboxes are unchecked, but you can leave them on. There's no ramifications from that. But what you really do want to set up is to uncheck the restore R data into workspace at startup, and then save workspace on exit to never. What this ends up doing is if you end up loading or you end up using R and RStudio The next time you open it, like after your computer restarts, or if you close it and turn it back on, it actually turns back on like with nothing loaded. And so there could be scenarios where like something's wrong with your R code. You want to essentially do the turn it off and turn it back on thing again. But if you have those, if you don't have the workspace set up this way, when you turn it back on, it's just going to load up all of the problems that you just had. So this way it sort of forces you to have a clean setup. The other thing is one of these days, or maybe right after this workshop, you're going to say like, hey, Excel literally can't open up this data set because there's too many cells, which is also a thing that can happen in Excel. So it's a really big data set and you can load it into R, but the only problem is the first time you load it, it takes like minutes to load, which is fine when you're doing the analysis for that project. The downside is if you are working on multiple projects and you close RStudio and you turn it back on again, it's going to take minutes for it to just turn on because it's trying to reload everything back to the state that you left when you turned it off or exited out. So this way, if something were to go wrong, when you close everything and turn it back on, it's actually starting off of a clean slate. And then I usually keep these two things also unchecked. So like none of the files that I have been working on also are open, and it's like a visual cue like, hey, this is actually empty. So that is the set of settings that I highly recommend and might save you a lot of headache in the future. Okay. So we're going to go through the process of loading in a data set into R. And before we go and download the data sets from the internet, there's a few things that are really important. Essentially, like how do I find the data set on my computer to load in? From the R perspective, we are going to be using a set of packages called tidyverse. This is a collection of R packages that are maintained by the folks at RStudio. And essentially, these are a collection of R packages that work with data, but they also work really well within, like amongst each other. So depending on if you've taken an R class before and depending on who your instructor is and whether or not the instructor learned how to program in R after 2015, you might have a different trajectory on how you're programming. But this is what I find is the easiest way to start learning R. After a certain point, things sort of just, there's a point after the introductory setting where you'll see a whole bunch of random different ways of doing the same thing, but hopefully this gives you like a more clearer introduction to everything. And tidyverse is really nice because all of the functions, because they are made to work with one another, they're super consistent. So you don't have to worry too much about like the ordering of how to use different functions in the tidyverse world. Okay, so we are going to end up downloading a folder that has all of the data that we're going to work with. So we're going to have this folder. I'm going to say like it's on my desktop, and there's a folder called ds4biomed. In reality, it's going to say ds4biomed-learner on your end. But if you are on a Windows machine, this folder on your desktop has a path. Like there's a location that the computer knows where this folder is. And that's going to be something along the lines of C drive, users, your username, your desktop, and then the folder, if that's going to be on your desktop. On a Mac, it's going to be very similar. Instead of the C drive, it's going to be a / and then it's going to be users, your username, desktop, et cetera, et cetera. Probably something you haven't really thought about because most of the time we are looking and exploring our data through here. But you'll notice that usually if you, for example, I click on my desktop, at the top somewhere, it might give you the actual path to that folder. So there is this notion of a working directory when you are programming, and the working directory is essentially where is your starting point. So we are going to set it up in such a way where if we are going to find this medical tumor growth data set, our starting point is going to be that ds4biomed folder. And so there's two ways from our starting point we can refer to that data set. One is a relative path. So like assuming our starting point is that ds4biomed folder, we can then just say the data set is in data/medical data tumor growth. Another way we can point to this data set is specify the entire path, which is from the very beginning. So if you've worked with other people who've used some kind of programming language before, depending on their understanding of working directories and how to run their code, you might end up like, hey, the data, here's the code for my analysis. You have to like sort of change your name at the very top. And that's because they're specifying their data set from their full path versus something relative. And there are conventions, like if you end up collaborating with multiple people, you should probably refer to everything as a relative path. But that also means that everyone in your group has to understand where the starting point is. But the nice thing with the relative path is I can move this folder all over my computer. And because everything is specified relatively, it doesn't matter like if I decide to move this folder to another location versus absolute paths, like you have to change something. So that is the benefit of doing that. One other thing about working directories and relative paths. Let's say right now my working directory is in a folder called analysis. How do I then point to the medical data tumor growth data set? So we can do this also relatively. And the way we say, or the way the computer understands, hey, go one folder up and then double click down the folder structure, is computers will use a set of two dots to say like, go one folder up. So go to my parent directory and then go down my, and then go down the other file structures. So if you need to say like go up two folders, it will be .. / and you'll just keep appending that. And the other way is you have to specify it from the very beginning, and then you can point to your data set. So there's the relative and absolute path is something that is really important when you're trying to find, like, I'm trying to load up a data set, and a lot of times people are like, oh, I took this R class and I'm going to do it on my own stuff. And then the problem is I get this file not found error and you just stop, right? And that file not found error is almost always either something to do with pathing or you type something wrong. All right, so as an exercise, you can either write it in your head or write it on a piece of paper. Given the following example folder structure, where we have a chart review project under the projects folder, and also a totally separate project that's an RCT, a randomized control trial, there are patient data sets in both of them. Assuming that we are in the analysis folder here, what is the relative path to the RCT patients data set? And then what is the absolute path to the chart review patients data set? And I guess if you are following along in Zoom, you can type that in to chat or something. All right. So if we want to answer the first question, which is what is the relative path? If we are starting here in the analysis folder, we need to go one folder up. So it's .. And then another folder up to the projects folder. And then from there, we can go down into RCT, down into data, and then down into patients. So that's why relatively we specify .. because we're going up 2 folders and then back down. Versus the absolute path, and this is the path, this is the solution. If you were on a Mac, it will always begin with the /. So if you ever see something that's like, this is where my folder or my file is, and the first character is a / or a C://, they are specifying a absolute path. And then if it's anything else, usually if it begins with .. or .. or like some other folder name, they probably are referring to it as a relative path. And so that is how you can visually check to see what's going on. The one weird thing with Windows, especially if you end up copying and pasting, or if you end up with people who work on a Mac, in Windows, technically you use the / for different folders. My suggestion to you is don't do it the Windows way, do it every other computer in the world's way, and use the / if you're in R programming and trying to specify different folders. If you can use the Windows notation, especially because if I were to point to a data set, I can copy this location up here, and that's useful from Windows. But if you copy this, the / actually means something really specific in every programming language. So if you want to copy paste stuff from the Windows Explorer, you have to put an extra / in there. And so that's one of the annoying things that you might encounter working between Windows and every other operating system. Or you can use the / and then don't have to deal with it. The downside is you can't actually open up the Explorer, copy the actual path from there. Either way, you'll either have to type an extra / or replace one of the slashes. All right, so right now we will go and download our data set. And you can do that. Hopefully you've done it already. And give me a second. There's literally 3 computers in front of me. Okay. So if we go to this website, this is where the data and all of the course materials are going to be stored. For you to download the data set that we are working with, you can click on this little code button. And this essentially applies to GitHub in general. You can download the zip file. It will pop up with the ds4biomed learner zip file. I am going to put it on my desktop because it's sort of easier for me. And then the actual thing, and people usually almost always forget when they do this, is when it's on your desktop, actually go and extract the contents of this folder. Computers have gotten useful to the point where they're no longer, they're sometimes being not helpful anymore, because the zip file, you can still double click and it'll open up like as another folder, but it's still like a compressed file. So actually go and make sure you uncompress the folder, because you will find out really soon there's going to be a point where like you can't actually save the file that we're working on, and it's because it's not uncompressed. So that's what you want to look out for. The next thing about setting all of this up, in our studio, in the top right-hand corner, we have a projects panel. After you've unzipped it, you can click on that, go to open project. navigate to the file that you unzipped. So it's, for me, it's going to be ds4biomed-learner on my desktop. And then you'll see this little icon that says rproj. And if you double click that, what you will find is RStudio will reload. And then in the top right-hand corner, it's going to open up the name of that folder. The other thing that you'll notice is the files panel in the bottom right-hand corner also opened up to that folder. And that's really important because we are working with projects. There is this really helpful function that says get my working directory. So we talked about working directories. Because we worked with RStudio projects and because it shows up in the top right-hand corner, this is its starting point. And that's why RStudio projects are really useful. It's because it can set up the starting point for anyone. So like, even if I can literally give you this folder on a thumb drive and you can plug it in, and as long as you open up that project on the thumb drive, it will know its working directory and starting point is going to be somewhere on that thumb drive. And so you can move around this folder. You know, maybe you'll have a folder on your computer that's like class, and you'll just, after the class, copy it over there. As long as you open up that project, all of the code should work again, assuming you are typing in relative paths. All right. All right. So that's one of the things that I want you to take away is how to set up your working directory. That's what we just went through. We don't need to do those exercises. All right. So the next part is actually going about loading up our data. You can see here, there's a folder called data. And if we click that, it'll open up the data folder. The one thing you do want to keep in mind is just because you are clicking around and exploring your computer in that file view panel, if you look at your working directory, it's not changing. So that is just for you to figure out, like, did I type something wrong? Like, it clearly exists when it's not working. But your working directory is going to be defined by where that project folder or project file actually is and like how you opened it up. So I talked about running code in RStudio. It's not very useful if you have to retype everything from scratch every single time. So what we can do is in the top left-hand corner, you can either click on File, New File, and then go to RScript, or there's an actual like plus with a new thing, and you can click on RScript. You'll notice a lot of times they'll give you the keyboard shortcuts for it. So if you want to learn the keyboard shortcut, you can. I typically try not to because I have to teach this stuff all the time. So once you open up a new RScript file, you get 4 panels. The top left-hand panel is now the code that you can type. So if I say 3 + 3, I can then select that block of code I want to run or put my cursor on that line that I want to run. And you can hit the little run button, which says run the current line or selection. And the keyboard shortcut for that is Control and Enter. Or if you're on a Mac, it might say like Command Enter or something. So whatever that thing says, do it. So I can either select the part that I want to run or execute that individual line using Control Enter or putting my cursor there and clicking the button. You'll notice it will take the code that I wrote up there and then copy paste it into the console and then run it from there. And so that's nice because now we have a way to take code and save it instead of rewriting everything from scratch or, you know, copying, pasting, or typing code in a Word document and pasting it back or something like that, which has been done in the past. So what I'm going to do is I'm going to save this file. So you can hit Apple S or Control S to save. And we can save this file as like 001 dash like first_r. Right. So one good thing about good practices as you start working with data is number your scripts in the order you expect to execute them. For example, you can have a script that says like clean.r for cleaning your data, and then another script that's analysis.r for analyzing your data. But if you look at your folder structure, A is going to be sorted before C, and it's like up to you to either take notes or recognize that like you have to go look for the C file first, run that before you can run the A file. And that just gets way worse if you have 10 files in there. So literally just put a number in front. I suggest you use a 2-digit number just because computers get really weird when you get to 9 and 10, and it might just start numbering things like 1, 10, 11, 12, 19, 2, 20, 21, 22. So computers are kind of weird that sometimes they don't recognize things are numbers. So use a 2-digit numbering system. And then that way, if you open up your project or someone else opens up your project, they know exactly the order of things that need to be done without having to guess. Okay, so that is the first hour and 8 minutes. Let's take 5 minutes. We'll take a 5-minute break. And then if you have questions, you can ask questions, and then I'll hold on like our specific questions until we come back from break. So it is right now 8 past the hour. So 5 minutes would be 13 past the hour. So we'll just come back at 13 past the hour. Oops, sorry. Okay, so a few things that happened on the Zoom side. If you go to the actual course website, under setup, there's a section called binder, like literally like for installation issues. If you want to go through this material and for some weird reason, like you're on the library computer or some other random person's computer, or you want to make sure that stuff works, why stuff not working on my computer, but I want to make sure it works on like another setup, you can go and literally click this launch binder icon. And if you give it about 15 seconds, or depending on when the last person opened it, sometimes it'll take 30 minutes. It will open up R and RStudio with the data sets in your browser. So you can actually go along and code through everything that we're going through today. One thing you'll notice, and it won't matter in today's class, is the version of R is going to be different. And for a lot of the stuff that we're doing, this really does not matter. So don't worry too much about versions of R not matching up. The one thing you do want to keep in mind, if you are using binder and you end up saving your code and stuff in here, is there is, I don't remember the exact timeout, but if you were to like go on a 10-minute break and come back, binder is going to close. And if it closes, or you close the tab, or you do something weird, it will not persist any of the stuff that you worked on. Meaning that all of this stuff, if you've just like done an entire analysis somehow through binder, and if it refreshes or something, you lose it. So how do you go and download stuff off of binder? If you end up, you can use the little checkbox to check off what you want saved. And then under the bottom right-hand panel, you can click on more and then export, and you can download the contents down to your computer. So use the binder URL if you end up falling behind, or like something horrible happened with your R and RStudio instance. Also, if you end up needing help in the middle of the workshop and I can't help you during a break, like I'm just gonna say like use binder and we'll deal with it like later on, just to keep the class moving. But this will have the data sets and all the packages and everything you need set up for you. Just it doesn't persist any of the data or files. The other question from chat was, we talked about numbering scripts. I suggested using a 2-digit numbering system. You can get, what I actually do in my actual day-to-day is I use a 3-digit numbering system, and this was also suggested in chat. And instead of counting by ones, you count by tens. So instead of 0, 1 clean data, it'll be 0, 1, 0 clean data. And that's because inevitably you're going to end up in a situation where like you're counting 1, 2, 3, 4, 5, and you want to shim something in between 2 and 3. And so now you have to go and renumber a whole bunch of your files. So if you end up counting by tens, you can literally put in, to put something between 2 and 3, you can put 0, 2, 5, and then it will sort properly and you don't have to rename any of your files. So I've done that many, many times. And I typically want to say, if the person who suggested it, you probably like seen the Linux file system at one point in your life, and that's sort of like what they do as well. And that was like initially my inspiration of like changing my personal workflow for that. So yeah, so the main takeaway there is like, really just like number your scripts in the order you want to execute it. It'll make your life way, way easier. And if someone from like computer science world like makes fun of you for it, it's not compiled code. It's totally fine. It's for an analysis workflow. So totally fine, because it is more legible and you won't have issues of running things out of order. All right, so we're gonna go back to RStudio, and I am going to create a new script. I'm going to save this script, and I'm going to call this 02, and I'll call this load.r, or just load, and it will automatically append the .r file extension, like .csv, .pdf, .jpeg, et cetera, et cetera. One other thing when you're naming things from anything that a computer programming language will use is do not put spaces in your file names or your folder names. It is going to cause you more headache than it's worth. If you end up putting this folder into some part of your file system that has a space in it, like there's not really much you can do about that. What people end up doing is like the name of their folder on their computer is like first name space last name, and you have a space in the folder path. So that's also, it will get annoying if you have to deal with full paths, but as long as you keep things in relative path format, hopefully having spaces in your file names do not become a problem. You will, and then my suggestion is use a combination of dashes and underscores to sort of prevent having a space in your file name. It might look less pretty when you're like double clicking on your computer, but it will make your programming life way, way, way easier in the future. All right, so we are going to load up. I talked about tidyverse. So we are going to go and load up the tidyverse library so we can go and load up our data sets. So the way we do that, if you have everything set up, there is a function called library. It is a command that we can run. We know it's a command because it has a set of round brackets at the end. And then in there, we tell it the library we want to load. So I can say library tidyverse, and it will dump out to the screen a whole bunch of information. But as long as we don't have an error message, and that's different. R is a little weird. Sometimes red text will come up as a warning message. It will look like an error, but it's actually just saying like, hey, just so you know, not like a, hey, the code didn't run. So R is a little weird. Sometimes not all red text is bad text. It's or like error text. So that's something you want to keep in mind. All right, so I'm going to periodically or very constantly hit Control S. So like I'm going to be resaving this file throughout. So the next command within tidyverse, it has a function for us to load up CSV files on our computer. There is a function called read_CSV. And if you're in part of the world where your decimal separator is a comma instead of a period, then you will use read CSV2. But read CSV will read comma separated values. So if I hit enter, it'll automatically autofill or autocomplete the round brackets for me, or you can type it in yourself. And then in here, in either single quotes or double quotes, you can put in the location or the path to the data set you want to load. So you can use full paths, but I'm going to do everything through relative paths. So the data set we want to load, if we look at the bottom right-hand corner, I want to go into data, and then there's literally medical tumor growth.csv. So how do we point to that file? We literally say data/medical tumor growth.csv. The nice thing with RStudio is if you start typing bits and pieces of a path and you hit the tab key, which is the key to the left of the letter Q on most keyboards, it will try to autofill and give you the file structure so you don't have to type as much. And more importantly, you don't end up making a mistake in your typing. So if you end up using the tab key and you're like, wait, my data set like isn't showing up, it's probably some pathing issue or you type something wrong in the beginning. So I'm going to load up the medical data tumor growth.csv file. And now I'm either going to select this line or have my cursor on the blinking cursor on that line and hit Control Enter or hit the run key. And what you'll see at the bottom is R loading up the medical tumor growth data set. And this is a data set about different treatments and different days for each patient in some treatment arm, and then the size of a tumor. And I don't remember what type of tumor it is. I need to look up the notes on where I got the data set from, but this is tumor growth data. So you'll notice that at the bottom left-hand corner, it dumped out the data set to the screen. And it's not going to print out everything to the screen, because if, for example, this thing had 1 million rows, like you as a human being will not be able to comprehend 1 million rows of data, no matter how, whatever. So the reason why it's only showing you the first 10 rows by default is so the most important thing you want to check is that the column names are read correctly. And then maybe some of the values are being in the right column, because you never know with CSV files, or especially if they're manually curated, how things are working. You want to make sure that the column names are correct, because not every data set will have the column names on the first column, first row, or they might not have column names at all, and it comes from another metadata file that you just need to know that these are the column orders, et cetera, et cetera. So depending on where you got your data from, that's something that you need to keep in mind. The other thing is, remember when I said that the top right-hand corner tells you about everything that you've loaded up and saved into R, and it's currently empty. And that's because the code that we wrote right now, all it did was load up a data set and dumped it back out to you. We haven't really saved it to anything. And that's important because it's kind of not helpful if we have to reload this data set every time we want to perform an operation. So let's save this data set that we loaded into a variable or some object or thing that we can constantly refer to within R. So I'm going to copy this line of code. So it's still going to be this read CSV call, but at the very beginning, we want to load this into a variable. So we say tumor. So now I can refer to the data set as tumor. And then in R, if you've programmed in R before, you can technically there's a caveat with this statement, but in R, to assign something to a variable, you literally go and type less than dash. So you physically make an arrow. The official style guide also says put spaces around the arrow. So it's really clear it's less than dash the arrow and not less than negative number, negative value of something. And what this operator does, it's pronounced gets, is it takes the thing on the right-hand side and assigns it to the thing on the left-hand side. So tumor gets this read CSV data. If we run that, the top right-hand panel is going to load up the data set. And you'll also notice it gives us the number of rows and number of columns in our data. So this is really important. If you're reading in sensor data and it should be definitely 300 by 300, you should definitely see 300 by 300. Or you're looking up your metadata file and it says that this is a data set with 574 observations with 5 variables, make sure that actually got loaded up properly. On the bottom, you'll notice it gave you information about how it loaded up the data. It's not really that important right now what it says. For the most part, as long as I see it on the top right-hand corner, I essentially ignore whatever just got typed out because it worked. The other thing you'll notice is it didn't dump out the contents of the data set to the screen because we saved it to a variable. So if you want to see the contents of the screen of the data set we loaded, we now need to refer to it as tumor. We can hit enter and then we can see it on the screen on the console on the bottom. In RStudio, the other way you can look at this data set is if you click on anything other than this blue arrow, I essentially say click the thing that looks like a spreadsheet. RStudio will open up the spreadsheet view of your data set that you loaded. So now you have something that's useful and familiar and analogous to Excel in terms of just looking at your data. The only thing you can't do is actually go and modify and edit this data set, which is a good thing. You still have some ability to do filtering. So if I said something like, remember, like, give me all the values where like day is equal to 3, you can do that. This is sort of a new feature. I don't know if everyone has this, but you get distributions if you click on this, which is kind of like cool and interesting. But I typically just use this to browse around and look at my data set. If I like know an observation like exists, or like there's something weird with this value, like let me go physically go see it. That's what I will do. So that's loading up CSV files. Another common thing is, what if it's an Excel file? So we have to load up another library if we want to work with Excel files. The package that we need to load is literally a file called read.xl, and it will give us the functions that we need to load up Excel files. So very similar to read_CSV, if we load up read.xl, there is now a function called read_Excel. And the function looks very similar to read_CSV. We want to load up something in the data column, and then all we need to do is point it to an Excel file. And then just like before, we can say tumor_Excel gets this read.xl function. And now if we run it, we got a data set that loaded up our data set in Excel format. That's great. One other thing, if you have your cursor right before the opening round bracket and you hit the F1 key, it'll open up the help panel in the bottom right-hand corner. And so if we scroll down a little bit, in read.xl, the first thing is the path. So we talked about paths. It's like, where's the location of this file? But you'll also notice that if you have Excel files with multiple sheets, you can load up really a specific sheet out of the Excel file. By default, it loads up the first one. So when I talked about in that very first example of having your 3 patients in 3 separate Excel tabs, that's totally fine. You just have to read in that data set with the different sheet 3 separate times. There isn't a way to say like, here's an Excel file, load up everything in one, and like load up all of the tabs, because not every single sheet in Excel is the same exact data in the same exact format. So there probably is a way to load it, but it ends up being given to you as like a totally separate data object. And so it's just better to be really explicit on what sheet you end up loading. All right, so next thing, if you ever get confused with whatever is going on, like I saved this thing into an object, like what is this? If you're trying to ask for help, one of the things that you want to be able to know is like, what is this thing that, like, what does R think this object is? There's a built-in function called class. And if we give class like any like variable or the object that we saved and we run that, it will tell us what is that thing. So here it tells us it's a Tibble DF, it's a Tibble, and most importantly, like the thing to the last thing it says is called a data.frame or data frame. And if you see data frame, this is R's way of telling you, or most data science programming languages way of telling you, you just loaded up a tabular spreadsheet data, and that's what this is. So a data frame object, and you'll hear this a lot, is this is some rectangular data set that you just loaded up. All right, so I want to take about a couple of seconds just to make sure that everyone is caught up. So hopefully in the top right-hand corner, you have loaded up the project that we are working in. And then when you ran library tidyverse and library read.xl, you were able to load up this medical tumor data set. All right, cool. All right, so next thing, you're given this data set. There's a few things that you might want to do. First thing that ends up being really useful is I loaded up this data set. There's 5 columns. Sometimes you might load up a data set and there's 100 columns. If you're working with GWAS data, it's like 1 column for every SNP. And it's just like, I only care about 3 of these. So how do we go about selecting a subset of the columns in our data? So we have the tumor data set. And if I just run tumor and hit Control Enter, it will tell us, it will just dump out the data set to the screen. In tidyverse, there is a function called select. Select will select columns out of our data set. The way the select function works is the first thing you tell select is, what is the data set you want to select from? So we say tumor. We're going to pass this function a comma delineated list of columns we want it to select. So we'll notice that in our tumor data set, the column names are all beginning with a capital letter. So that's important because it will be case sensitive in terms of how we select our columns. So if I want just the size column, I have to remember to put in size with a capital S. If I hit enter, what gets returned is just the whole column that's just size. And if this is the subset that you want, remember, then you would essentially go back and then use the assignment operator and like assign it back into something that you want, right? So you might call this something like tumor underscore size, because computers don't understand spaces, or spaces mean something really important in computers, to computers. So if this is all you want, like this one column, and you don't care, it's split off by different treatment groups or whatever, you just want this one column, there you go. So what happens if we want multiple columns? So we say select. And then all we need to do is just add in by comma. So the first thing we put in is the data set we want to select or filter our columns with. And then type in the columns that we want to keep. It doesn't have to be in the order that the original data set is loaded in. So if I really wanted like 2 columns of group because someone down the line wants 2 columns for group because they want to do something with it, you can specify group twice. If things are out of order, you can re-specify the order, et cetera, et cetera. For example, if you're working with GWAS data and like all of your identifiers at the end of the data set, totally specify them in the beginning, and then the 3 or whatever number of SNPs that you want to look at, and then you can have your one data set. When you are working with tidyverse code, or if you end up Googling for help, or if you end up watching like through Twitch and live streaming people, and people who live stream their code, you'll notice that they don't always type their code in this manner when they are writing tidyverse code. So this line of code here, this select tumor group day size, is exactly the same as this statement. This is the piping operator. The way you pronounce it is then. It is % greater than %. So hold the shift key and then bounce back between 2 keys on your keyboard. And what this does, if we compare these 2 lines of code, they are functionally equivalent to one another. The way we read this is tumor, then select group, day, and size. And essentially the pipe operator takes the thing to the left of the pipe and puts it in as the first thing to the next object. So it's like, if you think of pipelines, it's saying like, hey, I'm taking your tumor data set and I'm passing it off to select, and select gets that object and then does the rest of the things with select. It's a little bit like weird right now, but it becomes really useful because we can take the output of this, which is this filtered version of our data set, and pass it into something else. So you can create these literal like code pipelines within your data. So that's a small intro to selecting data, the columns. The next thing that's going to be really useful is how do we filter our rows? And so the way we can either subset our rows is there's a function called filter. And what's nice about tidyverse things is a lot of the syntax is really consistent with one another. So filter behaves a lot like select, where the first thing that it takes is the data set we want to filter. So we can pass in tumor. And then now we specify some kind of condition that we want to filter our data sets with. So let's say, for example, I only want the data from my original control group, which is group where the group value is equal to 1. So here I can say group with a capital G because it's the name of the column. And then double equal is the programming way of saying like evaluate this condition. Single equal typically means take the thing to the right and assign it to the thing to the left. So double equal is a condition. So we want to filter our tumor data set such that group is equal to 1. And now if we run that, you'll notice that we have 97 rows instead of 574 rows. And this filter data set is just our control group. So if you are trying to work with data where you're manually curating things and you sort of like input your data in chronological order, not by like treatment group order, you can then just say like, okay, I curated this data set. Let me filter by my treatment group of 1 and then save it out. And then I can just do some analysis either in R or in Excel of just the control group data. And just like using the pipe operator with select, we can say tumor, then filter group is greater than 1. So these two lines of code, again, are functionally equivalent. And we'll see like, and it'll come up again, and then hopefully we'll see like why this ends up being like more useful. All right. So I'm going to copy this block of code again. Let's say we want to filter on multiple conditions. We want to filter the values where group is equal to 1 and where day is equal to 0. So all we need to do is put in a comma and then say day is also equal to 0. So this is an AND operation. So both of these conditions for a row have to be true. And if it's true, it will give you back that data set or give you back that row. So essentially this is saying, I want to look at the initial tumor size for all of my patients in treatment group 1. And if I run this, I get a much smaller data set. Right now it is 8 rows by 5 columns. So there are 5 patients in my treatment group of 1. So our control group has 5 patients where there is a reading for the first day. And maybe that's something that you either want to make sure that like I really expected 8 people, hopefully I got 8 people. And this is one way you can go and visually check your work. And this is one of the benefits of using a programming language because you have some marker of like, this is 8 and you don't have to go manually count them if you are also using programming as a way to like check to make sure your data set is correct. Another way you can specify the AND operator is literally go and type in the ampersand sign. And this will say like group is equal to 1 and day is equal to 0. And the reason for me showing you the ampersand sign is this is the type of syntax you need if you want to specify or. So if I take this block of code again, and I turn this ampersand to a vertical line, this is called the pipe character from Unix world. Now I'm like trying to think like, what is this actual character in like symbol form? But the vertical line, which is usually shift and then the key above the enter key on a US keyboard, is the pipe operator. And this is how you say or. And so this means that either of these conditions can be true and I get my data set. So you'll see here, I get 126 values instead of 8 because I'm also, not only am I trying to capture values where group is the treatment, the group is 1, but it's also where day is 0. So I'm getting all of my control groups and then everyone else, their first day or their 0 day or their starting baseline value. And so that is how we can use or. And then now between using select and filter, we can now do a bunch of combinations of like taking a subset out of our data set. So if I can say tumor, then select on group, day, and size. And then remember, this outputs a data frame object or a data set. And then we can pipe that into filter. And we want the filter where, size is greater than 2000, for example. And so this is the benefit of using this piping operator because it makes it a little bit easier to read because we can take our eyes and then look down a column. What is this doing? It's taking tumor, then passing into select, and then passing it into filter. So we know very quickly what is generally happening to our data set. And if we care about some of the details, then we can scroll our eyes to the right. And this is essentially the difference between if you've ever gotten annoyed with like math notation where it's like f of g of h of x, like it would have been so much nicer if the code read like x, then h, then g, then f. And so that is sort of the nice benefit of using this piping notation. And it's trying to aid in making your code a little bit more like legible so you can more quickly figure out what's going on. So these two things are, again, functionally equivalent. It's sort of like, which one would you rather like read? Days, hours, months, minutes after you typed it. So, all right. So we've covered filtering, selecting our data. How do we start the process of building data pipelines? The easiest data pipeline is you have input data, it goes into a script, the script literally saves out a file, and then that becomes an input for either the next script or someone else, right? So that is, if you've ever heard about people talking about like data pipelines and blah, 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 et cetera, most of the time, and especially in the academic context, they're literally just passing around CSV files or Excel files from one thing to another. And then more cynically, it's probably an Excel file that's being passed around like through email and not like any other like reproducible way. But I'll show you the process of literally saving out this file. So first, let's create a data set that's a filtered version of our tumor data. I'll call it filtered. I'm gonna assign it something, and it's gonna be tumor. Then I'm gonna select a couple of the columns. So I'm gonna say group, I wanna keep group, I wanna keep ID, and remember the case matters. I wanna keep day, and then size, And if I spell select correctly, so I have those columns. Then I want to filter such that the day is equal to 0, and then, or the day is equal to 15. So let's say I only care about all of the values across all of my patients in all of the treatment groups. But I want to look at their starting tumor size and then like 2 weeks later, like essentially mid-month down the treatment plan. Right now, I'm only comparing those two values because maybe I'm only interested in seeing like, hey, are things better 15 days later? So now if I run this, because I use this assignment operator, like all the other examples I did, like I didn't use the assignment operator, but now because I did, I created a new object called filtered, and it shows up on the right-hand side. And so that's the other thing you want to make sure is if you are expecting something to be created, like literally go and make sure the thing got created. If it didn't, look down here to make sure it didn't error out, or maybe you forgot to actually put in the assignment, et cetera, et cetera. So look and rely on this environment panel. I do rely on that when I'm doing my own work too, because it's like, if I'm trying to filter on rows, I definitely expect fewer rows and I don't expect the same number of rows. And if that's the case, then I either made an assumption about my data that doesn't exist and that's why it didn't pass and the filter, or I typed something wrong. All right, so how do we go and save this thing or create like a data pipeline? So in tidyverse, remember we used read_CSV. Well, conveniently, there's a function called write_CSV. And all write_CSV, it needs to know two things. First, it needs to know the object in R that you want to write out as a CSV file. So right now, I want this filtered object. And then in double quotes, we put in where you want to save it. So this is where we go back into file paths and absolute paths and relative paths. You can save it anywhere. I'm going to say, well, there's a data folder in my data set in my project. Let me put it in the data folder. So we're going to put it in data. We're not going to select an existing data set unless we want to overwrite something. So we want to create a new data set. It's going to be called tumor_filtered and then .csv, because we actually have to be really explicit and put in the file. I want to say suffix, but that is the wrong technical term. The file extension. We have to be explicit in the file extension. And then another thing about computers, file extensions are, they don't mean anything and they're made up, and it's sort of like the whose line. It's just made up. Like you can, if you want, for example, like this is a really special file and I want to sort it, like you can literally call this .pizza and it will save as .pizza. The only thing about file extensions that computers use is there's usually a convention for these things. So when you double click .csv, it will try to open up a program that knows how to handle CSV files. So if I could, in theory, for example, name this .pdf, when I double click it, it'll try to open up the PDF viewer to it, but it's not going to work because it's not a properly formatted PDF file. So that's a thing, just so you know, sometimes proprietary software like does that, like they just create their own file extension. And so like when you double click it, your computer doesn't know how to open it, but it's like sometimes it's a plain text file. So that's like one way sometimes people like obscure things because again, file extensions don't really matter. So if we run this, write_CSV, if we go to the files panel and you'll notice this little arrow thing, it'll refresh the files panel. Now you'll see that there's literally a file called tumor_filtered.csv. And so I can hand this off, email this out. That is a CSV file like any of the other CSV files that we have worked with. If, for example, you want to, and I don't know if I have this set up in the instructions. I definitely don't. It might be in the binder instance, but there is a package called write Excel. It's a separate package because the folks at RStudio try to like nudge you away from like writing Excel files and try to like get you from stop using your data as a visualization kind of thing. If you load up Write Excel, there is literally there's literally a function called write XLSX, and that function will behave exactly the same as write CSV. Just make sure that you, just make sure you change the actual file extension so the Excel file properly gets loaded up as an Excel file when you try to go and double click this. And if you sort of want a refresher on how to like install packages, it's in like the setup instructions. So I'll try not to like go through that for time. And so now that we save this file out, we can open up a new R script. Another thing about working in R and just good pipelining script behavior, you'll notice even though I opened up a new file, the environment panel is still there. It hasn't changed. So that means that if I'm trying to refer to a data set, the stuff is still there, even though that I haven't explicitly loaded up that data set in this particular file. So one good rule of thumb and good practice is if you are moving to a brand new script, you essentially want to turn off R and turn it back on again. That's really annoying if you have to turn off the application and turn it back on again. So if you go to session on the top right hand, the top title bar, and you click on restart R, or the keyboard shortcut is literally Control-Shift-F10. I hit this key a lot. What it will do is it will close the R session and then restart it. And during the restart process, because we had set up like don't reload things automatically, the top right-hand panel is going to be empty. And that's really important because that's how we make our pipeline scripts reproducible, right? It wouldn't make sense for me to have this new file that like works on existing data set when I haven't even loaded it up, right? So that's how we can start building data pipelines. So in our new script, we have to reload tidyverse again. I'm going to save this as another file called, I'll call this, I'll call this stats. And then here, all we need to do is say read _ CSV and just the same type of notation, data / and then I can say tumor _ filtered. And then we can go through, and now I've created a totally separate part of the analysis that only works on my filtered tumor data set. And now I can say like filtered, I'll call this filter _ pipeline just so like you can visually see there's a difference. There's nothing wrong with calling this filtered again. But this is essentially how data pipelines work. It's literally as simple as load in a data set, do a thing, save it, and then in a new script, restart, and then load it back in again. And this is really nice, especially if you're, what I use this for personally, especially if I'm trying to make like publication ready figures, is whatever my data analysis is, I literally just write out the data set I'm going to use to plot, and I have a totally separate script that is just the plotting function for that figure. And it has its own number, and that's great because if I need to go and update the plot, like, oh, can you move the figure legend like over or whatever, I know exactly where that plot is, and I don't have to run the entire data pipeline because I'm just loading up that data set that I'm using to plot. So assuming that the underlying data hasn't changed, it's a really good way to figure out and find like the parts that you need. And if you've worked with people who don't understand or haven't really used or have never been shown how like you can create these pipelines, you'll end up with like, here's the code for my analysis. And it's like 5,000 lines long with like huge blocks of white space to like section off like, this is the cleaning and this is the analysis and this is the plotting. That gets really annoying. And I used to do that until I realized like, wait, I can just save this out and it's fine. And then for most academic contexts, that is good enough. You're essentially trading off like, I'm going to be having a bunch of like these intermediate data sets that might take up disk space. That is the trade-off, but I think it's worth it if it just like, you can find things easier, because I'd rather trade off disk space, which can be solved by like a USB or like call Arc for like access to like a server or something. Then like, here's like 5,000 lines, go literally Control F and look for something that you like typed. So, okay. So one question I got was, what is it like? Is there a guidance when saving an object to a .rds instead of writing to CSV? So one thing I didn't talk about, so I typically try to save things out as a CSV file. There are also a lot of opinions about like CSV files are terrible and they're slow and they're not efficient. I want to say for most academic contexts, it's more than good enough. And if I can just get people doing that, I'll be very happy. And most analysts that you work with will be really happy. There is another function. If you load up tidyverse, there is a read_RDS. There's also like the base R version that's read RDS. And also write_RDS. So if you load up tidyverse, like all of this is available to you. An RDS object is technically not a plain text file, meaning I have to use R to load that data set up. I can't like open it up in Excel because it's an RDS object is a binary, like only really R understands this thing. The benefit of using RDS objects is by default, they're compressed. And because they're binary, essentially it's just a mapping to how it's stored in memory. File sizes are usually smaller and load times are usually faster. Write times might also be taking longer because it has to compress it, but RDS objects are typically R workflow related. If you work in multiple languages, you might want to look into a project called Arrow for, it's run by Apache, so Apache Arrow. They have a binary object, which is trying to be cross languages. So you can take this binary object, which is good because it's space and speed efficient, and then load it up in other programming languages. But for the most part, just save things out as CSV because you can literally email it to someone and they don't need to know R. They can open up the file in Excel. So yes, it's ARROW. So if you Google Apache Arrow, that is the whole ecosystem. If you are specifically working between R and Python, there is a library called Feather. There used to be a library called Feather. I have to double check to make sure that's actually like the new name of the project and that's the one that's working now. But there is like a subset of Arrow that is literally just made to be bounced around between R and Python. So depending on who you work with, what your workflow is, you can look into more of these efficient data stores, or for the most part, use CSVs or Excel because chances are you might have like someone that just ends up doing all of their work in that workflow. So you need to sort of like accommodate for it. Having said that, there is nothing wrong in this write phase of your script to write out like the CSV, the Excel, and then the arrow and blah, blah, blah. Like you can, nothing's stopping you from writing out multiple formats of the same data set because you might use it for your analysis, but it's also useful to someone else. So this is code. If you want to write out multiple types, you're free to do so. Just remember when you load and run the code again, you run all of it. So everything's up to date. All right, so we'll take another short break. What you can do during this break is, if I can, there we go. What you can do is, here's an exercise question. There is a data set called all.csv, which is essentially our spreadsheet example that we covered in the second slide deck, and go through these steps. Use the select function, select those columns, use the filter to filter out a specific person, and then save it out as a CSV file. So we'll take 5 minutes. So I'll come back at 8 past the hour again. And then we will talk about running descriptive statistics and some, we'll get into the world of like, so at least you like end the day with like, you can do something with the data set that you have currently on your machine. And we'll set everything up for tomorrow. And then there's a bunch of buffer time to make sure, so it's not really, I really hope it doesn't need to end at 5 o'clock. So just come back at 8 past the hour. And if you want, you can work on this. Okay, so the exercise was to read in this data set, do some selection of columns, and then filter some of the rows. It's technically a small mistake in this. So I'll do this at the bottom of this script, and then all of this stuff will be on the internet that you can look at. If something happened and you want to make sure that you have all of the code. So I will copy the text here. So the first exercise, the first question was read in the data set called all. So I'm going to make sure that I have tidyverse loaded. I'm going to use read underscore CSV to make sure that to load up the all data set. So I know it'll be under data and all.csv. And if I just ran that, you'll notice it dumped it to the screen, but nothing showed up on the environment panel. So I need to give it a name. Let's just call that all for now. Then it says select a couple of the columns and then filter on some of the rows. So all I need to do is say all, then select. And the rows I'm going to select is going to be called first underscore name, last underscore name, day, time, temp underscore F. One thing to notice, or one thing for you to remember, not everyone is going to understand that they need to put in data sets without spaces, not only just the file name, but more importantly, like not as the column name. So it might look pretty to have first name all nice and pretty, et cetera, et cetera. Or you might have something like even more insidious than that, where like, hey, I want this column name to be centered. So let me put spaces in front of a column name, just so it looks centered in my Excel sheet. And that goes down into the, this is a data set, not a visualization. Like, what are you doing? And so one thing, especially if you want to check, if you're like, hey, I tried to load up this like column called first name, like, and it says it can't find it, what is going on? There's a function called names. And if we pass in names, the data set we want, we get just a list of all of the column names. So this is useful if you're trying to figure out like, did I spell it wrong? If you see like extra spaces before and after, like that's literally the name of the column that you have to specify. So if you have columns with spaces or weird spacing, the way you can type that into the select function is if you wrap the whole thing around, this is the backtick character. So it looks like the single quote, but it's the key to the left of the number 1 on a US keyboard. And it's, I think 6 or 8 on a German keyboard, on the AZERTY keyboard. So you use the backtick key, which you probably before programming never used before, to like essentially like do a double quotation. So this is useful if your column names have spaces, or if it has like a weird thing that you just want to be like, I want this thing exactly. You can use backticks for. And it also works if you just want to put it in for putting it in sake. So select, we selected the column. So now that ran. So I'm going to run the next part of the pipeline, which says filter such that the person's name is Anya Warner. So then I can say pass this into filter, and it's going to be, I can say, because like I don't need to combine first name and last name, I can say, I can do something like first underscore name is equal to Anya, and then last underscore name is equal to Warner. For this particular data set, I could get away with just checking one of them, but you can also do that by checking both of them. And here I have Anya Warner's value data set. Once I'm happy with that, and that looked like it worked, I can save this into a data set called Anya. So ANIYA gets this data set. And then I can use write_CSV to save that data set out. The example that I had said to put it into a folder called output. And if I run that, because the output folder does not exist, I'm going to get this error message. And it's going to say, like, I can't do this. So sometimes these error messages are helpful. Sometimes it's not. You have to sort of read them in context. That's sort of like the head banging thing of like working and programming that I showed in like one of the earlier slides of debugging. So the context is I tried to save out a file and then I got an error, right? There's nothing wrong with this data set, right? So first check to make sure that, is there anything wrong with the data set? No, that clearly works. So it has to be something to do with how I'm saving out this file. Maybe, is it because like I spelled this wrong? Well, if I had spelled this wrong, I would get it like, does this, cannot find this function. So it's not, it doesn't seem like it's a misspelling of the function. So the next thing has to be like, I've specified this path incorrectly. And it's not that I specified the path incorrectly. It's the fact that like literally this output folder doesn't exist. And so I'm trying to save into this output folder that doesn't exist. So the way we literally go fix that is you can do this in RStudio, or you literally go to the folder on your computer, like right click, create an output folder, and then that will work, right? So just because you're programming, you don't have to do everything in a programming in context, like literally like just create a new folder if that's what you need. You can write code to like essentially like check to make sure this folder exists. And if it doesn't exist, create it. And then if it does exist, now go save it. You can do that. The other thing you can do is when you are creating these project folders, like just create one with the output folder already there so you don't have this problem, right? So there's always a programming solution to things. You don't have to be overly like worried that like once you start programming, everything has to be done through a programming language. That is definitely not true. All right. Last part for today. We're going to talk about running descriptive statistics. So I'm going to go back to my stats script, and I'm trying to figure out if I'm actually going to use this. Maybe we'll reload the data set. So I'm on a new like thought process or a new part of the analysis. So I'm going to make sure I restart the session, make sure everything is nice and clean. I'm going to run this part of the code again because it's essentially like restarted. And we have this tumor data set, just like how we subsetted it. All right. This is data, I believe. How did we save this? Now that I'm trying to remember. This is data such that the day is equal to 0 or the day is equal to 15 across all of our patients. So let's say I want to just calculate the average tumor size across all of the patients here. So that's the question. I want to calculate the average tumor size at baseline across everyone. So what is at baseline? It is day is equal to 0. So we have to, that is the question I want to ask. First, I need to make sure I have data, the baseline data. So that we know is a filter operation where day is equal to 0. So I now have 37 rows where day is equal to 0. Great. Now next part of the question was I want to calculate the average tumor size. So how do I go and calculate an average? Well, the first thing I need to do is actually get the column that I want to calculate an average of. So here's one slight difference. I showed you how to get a column, and that's using select, right? And this is how you can, this is what I use. I also use the output as a visual reference to sort of help me, like, what do I have? You can see when I used select on a single column, it came out as literally a single column of data, right? And this gets sort of like really pedantic and like small, minute different like meanings of things. But if I were to give you a bunch of numbers and I say like calculate the mean, you technically don't get a column of numbers to put into mean. You just get like a sequence of numbers to put into mean. So like this output of a column is not what the mean function needs. It doesn't take a column of numbers. It takes like literally a sequence of numbers. So how do we get the sequence view of this column? So instead of select, it is a function called pull. And you can see right away, like there's visually a different output like format to all of this. One, it looked like a column, a data set of one column. And this one, it's literally a sequence of numbers. The square bracket here is going to be different depending on like how zoomed in your computer is. But essentially, it's just helping you count. So like this one, it's almost always going to begin with one. And it's saying like, this is the item number 1 in this list. And this right here is like, this is item number 15. So if you're trying to figure out like what item position that is, you can say 15, 16, 17, 18, 19, 20. So this is item number 20. So this square bracket stuff here is just trying to help you count if you're just like, that looks weird, what position is that? And then you can use that to back filter on like, I need to look up ID number, whatever, because that looks weird. So now that this looks like a sequence of numbers, well, R is a programming language made to do data analysis. So it has a lot of these things like mean, standard deviation, like pretty much every single thing you can think of with statistics. Like a lot of that is already built in. So we can pass that into the mean function and it calculates the mean. Great. So this is the answer to the baseline values across all of my patients for however I subsetted this data set in the past. Like that's either, yeah, across everyone. I don't think I did this for just group equals 1. So this is for all of my patients in this data set, in this experiment or whatever. This is their baseline starting value. So now that starts to ask the question of, this is, this was a thing that like literally showed up on Twitter like a couple of days ago. I'm trying to figure out if, there's no way I'm going to be able to find this in a meaningful time. Yeah, okay. So I'm not going to be able to find the actual like meme version of it. But so whenever you're working with an analysis, there is something called Simpson's paradox. And this paradox is essentially saying, if I took my entire data set and I just tried to do an analysis on it, that dotted line shows a trend that's negative. But if I break it up into 2 separate groups, like the blue group and the red group, it's the opposite. It's a line that's like positively sloped, right? So that has ramifications for like, are people doing better or worse according to your treatment? And like overall, because you just incorporated everyone into this one raw statistics, it looks like they're doing worse. versus like if you just broke them up into separate groups, which they're fundamentally different groups, it shows that they're all doing better. And so like age category is sometimes something where you'll see this. Like drug treatment groups is something where you might see this, et cetera, et cetera. So whenever you know that there's like some kind of group being involved, you probably want to do your analysis on like a group by group basis and not just like, oh, I just fit the model on everything all at once without looking at any of the other variables. Probably okay if you want to just make sure your code works, but definitely not okay if this is like your analysis to make like some policy or some decision or whatever, some treatment decision. Definitely not. So we want to be able to look at, let's say, for example, I just did the analysis of the mean across all of my patients' baseline values, but we just talked about Simpson's paradox and like I have these different treatment groups. Maybe it's a good idea to look at like different starting values for like different treatment groups. So there is multiple ways you can solve this. The easiest way that using all of the information that you know now, easiest way to solve this is literally in the filter parameter, you say group is equal to 1 and day is equal to 0, and then calculate everything else. So you can literally go and copy paste and add in the group information, right? And so if I want to do this for group 2, there we go. I changed that value from group 1 to group 2, and you can see like group 2's average starting value is a little bit smaller. We haven't covered the statistics part yet, and that's really just saving it off for like tomorrow, et cetera, et cetera. So we can do this for group 3 and group 4, et cetera, et cetera, et cetera, right? So one thing you notice is it's a lot of copy and pasting. And luckily for right now, there's only 4 groups, right? So like I can literally copy this and say like 4. Fine, maybe copy and pasting this 4 times, not really a big deal. I'm also assuming that you know what the numbers are. So like maybe it makes sense, it goes 1 to 4, but you don't know if it maybe just starts counting by tens. The other thing is, what if there's 100 groups? This will get really annoying if all of a sudden like something happens and you have to make 100 separate changes, right? What if you're like, oh, instead of day equals 0, I want to do the whole thing, but with like day 15, you're now like copying, I don't know, 1, 2, 3, 16, 20 lines of code, including the white space, just to make that one small change, right? So there has to be a better way. And luckily, there is a better way. So the first thing that you can do before you decide if you think copy and pasting is the good way to go, you can take this data set, so this filter pipeline data set, right? I can just pull out the group column, right? So I just get the values for group. And there is a function called unique. This is built into R. A lot of these functions you'll eventually learn as you use R. So it's not always like, it took me a while to know that like a lot of these functions exist. So build up on it. And all unique does is like, given a sequence of numbers, just give me the unique values. And so this is one way to figure out like, hey, is this copy and pasting method gonna be more hassle than it's worth or whatever? But I talked about a better way. And that better way is probably something if you know a little bit more of the buttons around Excel other than like just looking at things in Excel, there's typically a data ribbon or whatever they call it now. And then you can set up like a group summary statistics system in Excel. R can do exactly the same thing. So the problem that we're having is we want to calculate the average tumor size at baseline, so day equals 0, but for every unique value of group. So we have to say, take our data set, filter such that day is equal to 0, right? So that part isn't anything new. We want to do something for every unique value of group. And I'm like saying the sentence that way because hopefully that like, it helps you think about how you can structure your framing, and it helps point to the code that you need. So another way we can say every unique value of column or columns is group by, and that is literally also the button in Excel. So we can say group underscore by group. So if I were to run this bit of the code right now, it's not going to do anything, but group by is one of those interesting functions in that it understands that if you start making a calculation now, it knows to break out your data set into unique values or combinations of this column, calculate that data set and put it back together, right? So it does 3 things all in one go, assuming you've made a calculation. So it's called split, apply, and combine. So it will split your data into its components, apply a function, in our case is going to be the mean function, and then combine it all together into a single result. The group by is typically followed by a function called summarize. Why is it called summarize? It'll make more sense when I show you the code. So I will summarize something. I'm going to call it a new column called average AVG size. And that is going to be the value of mean of the size column. And what this gives us is in one go, split. So it will split for each unique value of group, apply the mean function on the column size, and then combine it back together into a single data set. So this is a data frame object. So we can save this if we want. We can into a variable, and then we can export this out to a CSV or an Excel file. And then if we don't know how to do plotting in R, we now have a data set that we can plot in Excel or whatever Tableau or whatever program you need to plot in. But all your data is there, and it's just like, here's the 4 groups, make a bar plot, and this is the height or the value for each bar. There you go. And if I get more patience, I can just give you the updated data set, and then you can just run your bar plot again. So that's a really nice feature, and that's all done by literally this one line called group by. So if I didn't, for example, filter on day equals to 0, for example, I can also group by the day. So let's say I remove filter and I say group by group and day. You'll notice that it gives me every unique pair of those columns. So it's group 1, day 0, group 1, day 15, because that's what we filtered on. And it will calculate a totally separate mean for each of those combinations. So now you can see, like, between just group 1, you can see, like, hey, like the tumor grew, and that's a control group, and you can see, like, hey, the tumor didn't seem to grow as much, and then the tumor grew, and then the tumor didn't grow as much. And so this is one way where you can quickly get a sense of, like, what is even going on with your data set, right? So even before we hear about or do all of, like, the fancy statistics and stuff, a lot of times, especially in medical research, hopefully the results aren't really surprising, because if you've done the exploratory data analysis part, you should have a sense of what's going on in your data set, instead of just like, I'm going to fit this really complicated model and it's a surprise, right? So another thing we can do with this is we can talk a little bit more about the summarize function. The syntax is a little weird, because what is a summary function? A summary function, like mean, is a function where its input is multiple values, like a sequence of values, and then its output is a single number, right? So that is a summary function. Like mean, I take in a bunch of numbers. It doesn't matter how many numbers I put in. I always get one number back. So there are other summary functions. Usually with mean, you also calculate standard deviation. So standard deviation is also one of those functions that takes in a whole bunch of numbers and it gives you one number back, right? What is not a summary function? So calculating z-scores or centering and scaling, those functions take in a whole bunch of numbers and give you the same number of results or numbers back, right? So that is stuff like z-scores and center and scale. Those are transforming functions and not summary functions. We can talk more about, let me see if I talk about transforming stuff. No, that's next week. But you can calculate like a centering, scaling, or transformation function. It's a separate process, but summary is what's used for summarization functions. So what's nice about this is within the summarize function, we can specify other things. So there's in R, there's an SD function for standardize, for standard deviation. And if you want, I think it's probs equals 0.25. You can calculate, you can use the standard deviation function. There's actually a function called quantile. So if you actually want like the 25th percent quartile or Q1, you can put that in there. You can put 0.5 if you want the median. You can put 0.8 if you want the 80th percentile of your data set. So quartiles or quantiles, those are also summary functions. So if you want to get a data set where it's, you know, small summarization functions, you can do that. And all right. All right. So I essentially said like there's extra time built in for random things. So that's pretty much everything I want to cover for today. Let me pull up the slide for things tomorrow. Right. So slide for things tomorrow. I showed you descriptive calculations today. If you notice, there's like something kind of weird with the output of our data set, because like I have the like day 0 and day 15 and then the average there. So it's a little bit harder to pass it on to someone else because they can't just say like, here's the size column, let me just plot that set of values. So tomorrow we'll start with like defining what is actually like a clean data set, and then show you all of the data manipulation steps that you can use to literally pivot from one format to the other. So if you're inputting data in one format because it's easier, how do you get into a format that's made for a computer? Or if your computer just gave you data in one format, how do you pivot it to something else that you can use like in another context? So that's all covered in clean data. That also feeds back into the spreadsheet chapter. And then once we have a clean data set, we can then do all of the other stuff, which is like making plots and fitting like logistic regression, or really fitting any type of model. So, so yeah, we finished 22 minutes early. So there is like buffer for like if you have general questions on things, but with that, that's really everything for today. And then tomorrow we'll start off with like a small review of everything we covered today, and then we'll just get it straight into all of the other fundamental data processing stuff. All right. So yeah, I will hang around until everyone leaves the Zoom room or the physical room. But yeah, if you have a question, you can ask. I don't even know if I think I can hear you if you unmute yourself also. But yeah, if you have questions, or like now is also a good time if like just R didn't work. If no one else has like any specific R questions, we can like just go fix it. Otherwise, I will say enjoy the 20 minutes of your day back. But yeah, so if you have a question, you can either type it, unmute yourself, et cetera, et cetera. So one question is using these materials to teach. Yes. So this is all, it's all under a CC, I don't know if it's CC BY or CC0 license, but it's under one of the Creative Commons licenses. So you can pretty much take whatever you want out of GitHub, this repo, whatever you need. If you want to use it to teach yourself, use it to teach other people, use it to whatever, just give credit back. There's technically like the persona pictures, those are drawn by my sister. So I might have to put that on like a separate repo just so like the images are on another, like a more explicit license for images. And then all of the other images that you'll see are by Allison Horst. There is literally a GitHub repository where you can go and download all of those pictures. I did not draw those. Allison drew those, and she deserves all of that credit because she's also a data science educator, and everyone uses her figures to teach. So, but yeah, the actual materials that I have, you are free to essentially remix, reuse, just link to that however you need. Yeah, so if there's any installation issues, again, you can always ask me for tomorrow and for any time in the future. Click the binder link if you just want to quickly spin something up after the class. Those links are, it's using a service that's essentially free until they don't exist anymore. So you should be able to have some working instance of this set of materials in the future. Just know that you can't save the results unless you have to explicitly export them. And if it like refreshes or something happens, like you lose all of your work. But if you're just trying to like teach a really quick lesson to someone, like you can spin up one of the binder instances to show like, this is how this works, and without having them go through the installation process.